Hello, my name is Gulnara Tagirdzhanova. I am a PhD candidate at the Shprabila lab at the University of Alaprota, and my talk today is comparative genomics of free fungi in a lichen symbiosis. I wanted to start with a question. How does a lichen work? Every lichen includes multiple organisms. First of all, the main fungal symbiont, typically an ascomycete from the class Lichenomycetes, a unicellular phototroph. And as we learned recently, many lichen also include a suite of low abundant symbionts that can include both yeast and bacteria. And together, these symbiotic partners can form a lichen, which is a stable association with a recognizable body plan, its own role in ecosystems, and so on and so forth. But how does it work? The current model is that the alga provides the fungus with carbohydrates, and the fungus in turn creates a symbiotic body to host the algal cells. However, no one really knows how the low abundance partner fit into that equation. How do they relate to the two dominant members of the symbiosis? What do they take from them, and do they contribute anything in return? There has been several hypotheses made that they are mutualistic symbionts or that they are parasites, but until recently, there were really no evidence one way or the other. Why don't we know that? Lichens can be a tough object to study. If we wanted to do experiments on the lichen yeast, we wouldn't really be able to because the yeasts are unculturable. The Lichenomycete can be cultured, but it grows extremely slowly. And well, this is a problem, but it's not a problem unique to lichens. There are plenty of other systems and other microbial communities with unculturable components. A usual alternative would be metagenomics. Metagenomics is the study of sequence data from intact environmental samples. And metagenomics great for bacteria, but applying it to fungi can be complicated. First of all, fungal genomes are much larger than bacterial, and also they are way more complex. And that can create troubles for metagenomics algorithms. But then there is another problem specific to lichen yeasts. The problem is that the yeasts are present in lichen symbiosis in very low abundance. And that makes it hard for us to get enough sequence data from them, because any given data set would be absolutely swamped with the genomic information from the Lichenomycete and the alga. One of our goals was to overcome these challenges and get lichen yeast genomes. To solve the problem of low abundance, we used a method that we called cortic slurry method. We knew from previous research that the yeasts live in lichen cortex, and they are embedded in the extracellular matrix there. On this figure, the yeasts are shown in green, so you can see that they occupy the outermost layer of a Bryoria thallus. We used hot water to partially dissolve the extracellular matrix and release the cells into solution. After that, we concentrated the solution and used the resulting slurry to make a metagenome. After solving the problem of low abundance, we needed to analyze this very complex metagenomic data. And our metagenomic analysis included 3 steps. The first step is assembly. We took lots of short sequences produced by an Illumina sequencing machine, and we assembled them into longer contiguous pieces. Sometimes people use a metaphor and say that genome assembly is a bit like assembling a jigsaw puzzle. The second step is binning. The goal of the step is to disentangle the metagenomic data and extract genomes of individual symbionts. And if I wanted to extend the metaphor, I would say that dealing with metagenomes is a bit like you have a dozen of jigsaw puzzle pieces mixed together in one box, and you need to sort them out. And you have to be really careful to avoid outcome like this one. So how we did that? We used a database-independent approach. This means that we did not try to identify genomes by comparing our data to previously sequenced genomes. This wouldn't work for us because this way you can only extract genomes that are similar to genomes that are already known. And when we studied, there were no genomes from relatives of lichen yeasts available. So we had a little bit of a vicious circle problem. Instead, we didn't use databases at all. We relied on trends and patterns within our data. This included first coverage, which is a proxy of cell abundance. The idea here is that in a sample, a more abundant organism will have more copies of a genome sequenced. And we can use this information to separate low abundance and high abundance genomes. The second method is sequence content. So it turns out that different organisms and different genomes have different frequencies of nucleotides and oligonucleotides, so short words, occurring in their genomes. So for example, one organism will have more ACT fragments in the genome when AGT fragments, and the other genome will have it the other way around. And you can use this information too. And finally, we assessed the quality of the genomes. So that was a bit of a lengthy introduction, but now what did we find? In total, we extracted 4 genomes from the cortex-slurry method genome. 2 of them we expected to find when we started, and 2 were a bit of a surprise. The first 3 genomes in this list are from fungi. 1 is from the dominant fungus, the Gulnara Tagirdzhanova, and 2 from different lineages of lichen yeasts. One of them, Cyphobasidium, we expected to find because it has been already reported from Alectoria. But then there were another genome that we expected less, and it was Tremella. Tremella is a group of basidiomycete fungi. It includes many lichen-associated species, and until recently, all of them were considered as parasites because on the sexual stage, they produce gall-like deformations. However, it recently became clear that Tremella in its asexual stage is ubiquitous in Litharia lichen. However, neither the sexual stage nor the yeasts were reported from Alectoria sarmentosa, our study system. And finally, I wanted to mention that Tremella is related to human pathogen Cryptococcus neoformans. The 4th genome belonged to the Granulocellae bacterium, and related bacteria had been reported from lichens before. We wanted to know if the yeast and the bacterium whose genomes we detected are present in Alectoria consistently, or are they, just some random contaminants? For that, I had to put my genomic work on hold and get back to the lab. So we designed an experiment to test this. And this diagram will show you the result of PCR screening we made. I hid the data for now, so I can explain the layout of the plot first. We went to 3 different locations in Western Canada, and in each we sampled several trees. And from each tree, we collected 3 samples. Alectoria sarmentosa sample, another macrolichen growing adjacent to it, and just a bare tree bark. And each of the samples we tested for the presence of Cyphobasidium and Tremella and Granulocellae. The purpose of this test was to first check if these lineages are present in Alectoria consistently, and then check if they are specific to Alectoria, because we wanted to exclude the possibilities that they are just everywhere in the environment and not specific to lichens. And this is what we saw. Cyphobasidium and Tremella are present in Alectoria consistently, and every Alectoria had at least one of the 2. Mostly coming from lichen-specific clade. With Granulocellae, it was kind of the opposite. It was only once reported from Alectoria, but it was quite present in tree bark. Therefore, we concluded that Granulocellae isn't actually a part of lichen symbiosis. Our final dataset included 3 fungal genomes, 1 from the dominant fungus and 2 from the yeasts. Each of the genomes had quality comparable to what you can expect from a genome sequence from culture. We annotated each, meaning that we predicted the genes and assigned those genes with putative functions. Now we can get to a part where we'll look into the genomes to try to understand what they tell us about the biology of fungi. We looked into many different aspects, but for this talk, I wanted to highlight three aspects. First, we looked into genes related to secondary metabolism. We did it because a hypothesis has been made that the yeast might be involved here. This graph shows the number of secondary metabolism gene clusters in different fungal genomes. And you can see that Alectoria sarmentosa here, the dominant fungus of the lichen symbiosis, has way more secondary metabolism gene clusters than Cyphobasidium and Tremella. So we can hypothesize that the licheneromycete is most responsible for the secondary metabolism production. The second class of proteins we looked into were chymes. These enzymes are involved in producing, modifying, and breaking down polysaccharides. And we became interested in polysaccharides because they are the main component of the extracellular matrix I mentioned before. This matrix is pretty much the main structural agent in a lichen because it glues lichen symbionts together into the lichen shape. And also the yeasts are embedded in this matrix. And there's a lot to talk about chymes. I wanted to highlight just one example. Both the yeasts, but not the licheneromycete, have genes responsible for synthesis of glucuronoxylomannan, or GXM. GXM is a polysaccharide known from non-lichen relatives of Tremella. So for example, Cryptococcus, the human pathogen, GXM forms a capsule that helps the fungus to avoid human immune response. And this polysaccharide is also extremely hygroscopic. So Tremella fuciformis, for example, has a common name, the jelly fungus, thanks to this polysaccharide. And the moisture retaining properties of GXM are even used in cosmetics. So here you can see a skin lotion made from Tremella. And we hypothesize that maybe the yeasts produce GXM that helps lichen to retain water. We could not answer our burning question, are the yeasts mutualistic or parasitic? It turns out that genomic profiles are not really conclusive. First, we know a lot about plant fungal interactions, both mutualistic and parasitic, including the genomic perspective, but when it comes to any sort of fungal relationships, we know very little, and therefore we had very little reference point. And second, as we know from plant-associated fungi, similar genes can play a role in both mutualistic and parasitic relationship, which makes any sort of inference even harder. But then there is some evidence that I wanted to mention. First, Tremella has a profile of nitrogen oxytroph. It means that it cannot assimilate nitrates or nitrates, and therefore it has to get them from some living organism. Second, all three fungi have putative polyol transporters. And why is that important? Because polyols are the carbohydrates that the alga delivers to the licheneromycete. And third, Cyphobasidium is predicted to produce enzymes that target plant polysaccharides. Taken together, this evidence led us to hypothesize that maybe the yeasts might rely on alga for sugars. And this in turn can be viewed as evidence against the hypothesis that the yeasts are parasitic on the licheneromycete. To sum up, we used a combination of a wet lab technique we called cortex slurry method and bioinformatic analysis to obtain the first two genomes of lichen yeasts. When we started, we only knew two things about lichen yeasts, that they exist and what taxonomic group they belong to. But now with these genomes, we generated a set of testable hypotheses about the metabolic potential and the trophic strategy. And this is the first ever evidence on the role the yeasts play in the lichen symbiosis. And with that, I wanted to thank all collaborators and my lab mates and the funding sources, and thank you for your attention.